weekly Wednesday webinar training. Appreciate everyone being here. Today, the subject, we're going to have Yield Masters Solutions come in and visit with us about the FX product lineup with AgriVive and this unique set of products that we're offering and solutions in the microbial space. Why we like these products is they offer many different microbes that have activities that provide function in specific species of plants that they're in. So they've been created to give a base level of microbes that many different plants like, and then specific to, say, corn or soybeans, other species have been brought in to help target those and create good activity in those plants. So very unique products in the marketplace. And we'll have Josh O'Brien with Yield Master Solutions. We'll have Shandy Bertolino on here to help us. And then I think that Tony Hagan is joining us also as we go to visit on some of the comments. Tony's the creator of the products and makes the products for us. So Dan Johnson is our regional manager up in the north. John and Amanda are up in the east, and then Chris is working eastern Iowa, parts of Wisconsin and Missouri. So we have that team here to support us today for any types of questions or comments also. And then obviously we've got this good team to visit. So Josh, we're going to let you kind of introduce things as you wish. I'm not going to do a 15 or 20 minute introduction this time. I think that content is out there for everyone to see in the moment if they want to go back and pick that up today or tomorrow or whenever. And so we'll let you get going here in a second. For the group that's on, before we get into the meeting, might you have any specific topics you wanted to cover with regards to products that we're going to visit about ion effects, soy effects, and crown effects that we should get answered, or any specific comments? You're welcome to raise your hand, either chat with us or take yourself off of mute and insert yourself into the conversation if you would like. We'd like this maybe to be a little bit conversational as we can. We can all go on and on and on about products and features and benefits, and that's always helpful, but the interaction we find is good also. And it was kind of nice after our last meeting. Some of the people stayed around and we visited for another half an hour. So we'll make ourselves also available after the meeting. We'll shut off the recording, and then for anyone who wants to stay on and chat further for more discussion, we'll stick around for that also. So if anyone has not raised their hand yet, Dan, I think you're watching the chat session as usual. Reminds of FFA, right? Dan's stationed by the chat session, so he's the doorkeeper. We appreciate that. And Tony, thanks for joining us today. Also, at times wanted Tony to insert specific comments, so we'll prompt him here and there about how he's maybe come up with the development of these products, what leads him there. A little bit about his story to help fill in some of the other details that really brings the specialness to this kind of lineup that we're looking at. So thanks for making it in today, Tony, also. With that, if there's no further comments or introductions, Josh, take it away.
Yeah, no, thank you, Michael. I appreciate the opportunity, and thank you everyone for being on today. I know a lot of you are probably just getting back from the holiday session with the in-laws. Hopefully that went well. Nobody threw food at each other or anything crazy like that. So Today is going to be a lot heavier on the conversation than on the slides. So if you guys have specific questions, please jump in, ask them. My goal today is to have this be kind of just an open dialogue, as if we were all sitting around the coffee table at the diner at 7 in the morning, just chatting about products and having an organic conversation. So hopefully we can deliver that feel for you. So Brian, Michael, Dan, you guys also please jump in and ask questions as needed as well. Without further ado, Shandy, why don't you go ahead and introduce yourself first and your role and your territory, and then I'll jump into YMS, and we'll bring Tony on after that for his introduction.
Yeah, thanks, Josh. Shandy Bertolino here, Morrisonville, Illinois. I'm an account manager, sales rep in Indiana and Illinois. I'm kind of South Central Illinois is where I'm at. So if you guys need any help out in the field, that's what I'm here for. So appreciate the introduction, Josh.
No, thank you, Shandy. Appreciate that. So let me pull up the slide deck here, and then I'm going to bring Tony in just in a few slides. Tony, if you want to do a mic check real quick, just let us know that you're good to go and available.
Yep, I'm here.
Awesome. Thank you. All righty. Let me go up to
Tony, if you're in a place and you have bandwidth to be able to put your camera on, I'll spotlight you in the video also, so that people can see as you speak.
Okay, so let's go ahead and get started. YieldMaster Solutions, you guys have heard this wrap a few times. I think a lot of you have seen our presentations. Just a quick reminder, we don't make anything. Our goal is to discover innovative biological products like what Tony develops, and we then bring them to the marketplace. So we help them out with providing them a sales and marketing team and marketing messaging on how these products should work. And then the biggest, I would say, service that we provide with our partners is our proof of concept trials, which you can see a map of here for 2022, what that looked like. So we go and ground truth these products. Not that we don't believe what our manufacturing partners tell us, but we want to see it for ourselves. So we put them in a whole different host of different proof of concept trials, whether it be side-by-sides, whether it be strip trials, big plot, small plot, everywhere in between. So we get a pretty good look at how these products work year after year. And we're still learning each year, I would say. But if you're not learning in the biological market right now, then you're probably not participating. So it's exciting to be able to share the data with you. And we have some great data today to go through on the iron and the soy in particular. That data primarily comes from our internal plots. You can see they're scattered around the upper Midwest here. We do have a lot of customers, though, as you go further south in Shandy's neck of the woods into Indiana, Illinois. So don't be turned off if you don't see your state up here. We do have data from other locations, but these are the plots that our agronomy team are walking almost every week, if not every week, writing down notes and observations. And this is the data that we're going to show today, just because we know it's the most replicated and reputable data that we have in our system. Tony, why don't you give an introduction to yourself and your company before we jump into INFX? And if you don't mind, I think it's important for the group to hear a little bit on just how you manufacture products from your discovery process, how that's maybe unique compared to other products or other companies in the market.
All right, my name's Tony Hagan. I am the crazy scientist that goes out and finds these biologicals. I am a little different than most of the large companies that go out and find biologicals. I will be driving down the interstate. I look for plants that have a unique characteristic. They grow bigger, faster, taller, grow in the cold, grow in the heat. I'll be driving down the interstate, and if there's something cool along the interstate, I will slam on the brakes, pull over, grab the plant, and try and pull the biologics out that made that happen. So a lot of the biologics we have come from finding a plant that has a unique characteristic, something that, say, it survives frost. Every other plant along the side of the road is dead from the frost we had. There's one plant that's big, bushy, survived it. I will stop and grab that plant, throw it on my kid's lap in the back seat, and pull it back into the lab. tear apart the biologics inside of it, see if that's what caused it, and add that to a cropping system. Most of these products have been developed from that. It's going out in the field from relatives and friends' fields. Is there a particular plant that grew larger than others? I like invasive plants. I like resistant weeds. Is there something in there that gave them an advantage that I can take out and put into a cropping system? And that's kind of how most of these products, the genesis of most of these products.
Awesome, Tony. And then how long have you been making these products for, Tony?
Some of these products started in discovery all the way back in 1993. So there's been a lot of development that went into them. I'm one of those kind of.
Oh, Tony, sorry, you got muted.
There you go.
Sorry. Most of these products are a series of developments. If I find something that works over the last 5 years, we've been trying to see if we can make it work better. So the development into these became multi-stage biologicals. If I can find a bacteria I can put on as a seed coat that will benefit at harvest or post-harvest, we've done our best to get those in there over the last 5 years. Stuff is always in development here. We've got a whole freezer full of pipeline that, as we are able to overcome obstacles in crops, that's one of the things that we're working on here. If a person sees something that a particular genetic or variety of plant has for an issue, let me know. Let's see if we can find something that will help overcome it. Better lateral roots or deeper taps or something like that. We found that one product for our lineup was not effective on all plants. So we went species specific. We have products specific to corn, specific to small grains, specific to legumes. We even went down to a difference between dry beans and soybeans, because there's a different biochemistry inside the plant. And it's been an interesting journey, figuring out all of these differences within the plants and being able to impact them biochemically specific to a species.
Yeah, I think that's one of the really cool things about your product line is it's not a one trick pony that you put across all the acres and expect it to work. It's very dialed in. So we're going to go through 3 of them today: Ion FX for corn, Soy FX for soybeans, and then the Crown FX for wheat and small grains. And I'm going to kind of ask Tony to give a little bit of history on each product as we're going through it. I'll give you the tea leaves of things you can find in the literature and try to express that to you as best I can. But since we have Tony today, I think it's a good opportunity to get a little deeper and just know some of the history of these products. And I think I'm even going to learn a little bit on it today too. So
Yeah,
I'm kind of excited.
Tony, one of the things, if you would mention a little bit more, I think in terms of the when we visited early on, you brought up this concept that not all activity in the plant is done based upon just because of the genetics or just because of a chemistry we apply or just because of, say, a micronutrient or a fertilizer that we apply. So can you talk a little bit about how you figured out or went into that concept that potentially the biology made the plant grow faster or do something versus that it was already just inherent in that plant and because of the genetics?
So I come from a biofuels background, and the project I thought I had a plant that made ethanol itself. And it took a very smart microbiologist to convince me that the plant was just a giant bag of bugs. And She is an outstanding medical microbiologist. And the fact that we are 10,000 to 100,000 bacterial cells for every human cell in our body, it became pretty obvious pretty fast that she was right. And once I came to the realization that the bacteria in the plant actually impacted day-to-day activity, not necessarily just the plant's genetics, it became a lot easier to try and find things like herbicide resistance that's benefited by bacteria in there. Bacteria have a huge influence on herbicide resistance. That was a shock to me. Um, Impact on solubilization, impact on pH inside the plant. That stuff was regulated by the microbes inside of it. Just like our guts impact everything from our mood to our skin, uh, our metabolism, it does the same thing inside of a plant. And once she finally convinced me of that, and she beat me over the head with that for many years, uh, it kind of opened my eyes to, there's a possibility that that one giant plant out in the field might not be refuge in a bag. That could actually be, and we found this out. This was really cool. Sorry to get off on a tangent. That bacteria in there actually came from a waterfowl flying over the field, pooping on the plant early in its life. Because we were able to trace that bacteria back to an aquatic system. So it's kind of cool the way nature takes care of itself. And once we kind of dialed into that being the case, we had some really cool discoveries.
Wonderful. Yeah, that's amazing. Amazing. Amazing concept to me. And I like how you linked human health to this also. It is eye-opening when you read or see the data that shows that there's more non-human cells in our bodies than human cells by number, maybe not by volume, but by number. And they drive all kinds of processes. If we did not have live microbial activity in our own bodies, I would say we probably would be dead.
Awesome. No,
Josh?
this is good stuff, guys. This is great conversation here. So why don't we dive into it then? Let's jump into the first product here, which is Ion FX. And again, I'm going to give you the T notes, and we're going to have Tony kind of chime in here. But application wise, we've used this product as a seed coat primarily in our treatment plots. But Tony, correct me if I'm wrong, in furrow, as long as we don't mix it with chemistry, is a really nice option for it. So if you're going to put it in furrow, put it with either just water or put it in a dosatron tank if you're gonna go out there with starter and kind of separate the two tanks there. 16 ounce per acre rate in furrow with it. And then foliar, Tony, how early do we want to get it down if we use this product foliar in order to see the benefits of the product?
For a foliar product, you want to get this on as early as possible. A lot of our biologicals are focused on early growth and rapid development. So the earlier you get it on, the better. If you can get it on shortly after emergence, you'll start to see a difference very quickly on these. You want to shoot for about pop can high for the optimal application time. That's actually pretty cool to see when you can actually influence when you hit R1 by getting this on early. We influence reproductive rate, vegetative growth, root development. The earlier you get it on, the more benefits you get.
Yeah, and again, our experience with it has been at seed coat, which is nice. For us, it has a 100-day seed viability on it, so it's not like a standard inoculant that you're going to run out in 30 days or 14 days or anything like that. So it is a nice option as a seed coat if you have the capability of over-treating seed with it. But mode of action wise, Tony, ballpark. I know you never give us the exact number, but just give us a ballpark of how many different strains of bacteria you have in this product.
So the product for 2023 has 147 bacteria in it for Ion FX.
So 147 bacteria. So you guys can understand why compatibility with chemistry can be a little bit of an issue when you go foliar and in-furrow. The likelihood that some of those are going to be somewhat sensitive to chemistry is pretty high when you have that many different strains in the product. But Tony, we've talked about this quite a bit in the past. You know, you have the workers in the product, and then you have the support system in the product. Give us just a brief synopsis of what you mean by that. Obviously, all 147 are important, but they're important in different ways, and walk us through what that might mean.
So we have biologics that support the bacteria in the bottle. So we have some of those bacteria are designed only to live in the bottle to keep everybody happy. This is all based on community. Biologics need friends. It is a thriving community inside of the bottle. Some of the bacteria are just to keep it alive in the bottle, so you get the best quality of living organism when you apply it. We have other biologics that support it for as little as 3 hours on the leaf to get it in as a foliar application. Some of those bacteria are only there to provide support when it's exposed to oxygen. They will help they help open the plant's pores and open the waxy layer on the plant itself and keep it alive until it is actually incorporated into the plant. Once it's inside the plant, it will actually, most of these are mobile. They will swim and they will migrate to areas inside of the plant that support them the most. So some are anaerobic, some like a lower pH, some like a higher pH. So they will actually move within the plant's vascular system to the area where they are most comfortable. So once they're in the plant, they start to divide and they will move throughout the plant and then move daughter cell to daughter cell once they're established. So it's a great inoculant. It's like taking a probiotic for a human. Once it's in the gut, it will migrate in the gut to the area that supports its best growth. So they all want to live in their happiest place, and they will move within the plant to find that happy place.
Yeah, I think that's a great analogy, Tony, that you know, bacteria are a lot like humans, right? They want to survive, and they want to survive in a comfortable area. Although there's days I live in Minneapolis here, and it's 50 below zero with a foot of snow, and I'm wondering if this is really my happiest area. But yeah, they're bacteria are really fascinating in that way. How they kind of migrate up and down the plant to find their comfort zone there. So as we talk key benefits, and this is where Tony's going to chime in on the next slide, is just the heat and drought stress mitigation. We've seen that dramatically in our plots, and I have some pictures I'll show you here in a minute. Plant growth regulator response. I do want to be very clear that this is not a PGR product. We are just allowing the plant to maximize the PGR production of its own, and taking advantage of that. And then with that, then we see a lot of increased ear size, improved ear fill, and then the residue management is probably one of the coolest kind of freebies, I call it, with this product, that we don't really charge into the price of the product, but there's a dramatic benefit to it, and we'll go through that towards the end of the product here. But Tony, walk us through this kind of point by point. Let's start with root development and then the secondary brace roots. You know, how are we going to see that? Is that going to happen every time? You know, just give us a summary of those two benefits to start with.
Okay, so rapid root development, those fast laterals are going to be very important. So to maximize your banded fertilizer uptake, you want to get those roots to that fertilizer as fast as you can. The longer it takes the roots to get there, the more leaching you're going to have, the more loss to rain you're going to have. If you can get those roots to it fast and start to take up that nutrition, that's a huge bonus. It's all about that nutrient uptake and efficiency for that fast root. The secondary brace roots, those are from a very specific plant that we got from Central America. And those secondary brace roots are a function of that lateral root expansion. We maintain the pH inside of the plant throughout the day. Rather, we reduce its size of fluctuation. By reducing that fluctuation in the early root development, we're promoting stalk growth, and we don't get that pinch point at the soil line. By removing that pinch point, the side benefit and the visual cue that has worked is a secondary set of brace roots that you won't see on an untreated plant. So that secondary bracelet is a really good visual cue that you got this inoculated correctly. It is in the plant and working.
So Tony, if we go in fully with this, how early do we have to get on the plant in order to see that secondary bracelet benefit?
Up to V3.
V3, okay. Because I know that is a question we get quite a bit. But if you ask me about this product, and the way I normally present it when Tony's not here, is talking about the PGR response that it elicits the plant to make on its own. And I think, in my opinion, that's probably the biggest benefit. I'm sure we could debate, the other things that are happening, but to me, it seems like if you're chasing yield, the ability to take the governor off the plant and convince it that it's as healthy as it can be seems to be the biggest influence to that. But Tony, walk us through when we're trying to kind of, take that governor off and allow that plant to believe it's as healthy as it can be. How are your bacteria assisting us in doing that?
Okay, so in the ground, there are solubilizing bacteria. They'll solubilize iron, zinc, magnesium, calcium. They'll make it available to the plant. Once the bacteria get it into the plant, most of them lose influence. So pH is going to fluctuate a large amount during the day. There's a nightly reset that resets all of those ions back to a usable state by the plant. Our bacteria actually are actually solubilizing inside the plant. This was one of the biggest things we got to work is we maintain inside of the plant the biochemical happy zone. So there's a zone inside of metabolism where you're not winded, the plant isn't too hot, and everything works really well in this small zone inside the plant. That's why you can hear your corn grow at night, because the reset happens at night. So during the day, we're allowing that plant to stay in the happy zone of production longer during the day. And in the trials we've done in-house, we have this cabinet where we have heaters and extra lights and a photosynthetic meter on the plant with a pH probe stuck in the side of it. We actually watch how it changes the metabolism inside the plant. By keeping the plant happier, the ionic state of the calcium, magnesium, iron, and zinc in a usable state, so it hasn't gone out of production, that allows the plant to make the PGRs, to create those new cells. It gets pretty technical on the biochemical side, but easiest way to think of it is it keeps it fat and happy all day.
Yeah, I know in our plots, we've seen it all the way through, and those of you that grow silage, we'll get to that here in a little bit, but we've seen it all the way through where the bigger stalk diameter, the bigger leaf tissue size, obviously bigger ear set, it's just a lot bigger plant when you're using Ion FX, and I think that's probably another big reason those secondary bracelets are so important as well, is just supporting that plant as well. So it's really fascinating when you start walking fields that have Ion FX in it and comparing it to the check, especially, on those early kind of V6 through, V8, V9 stages of the plant life cycle, just how different they are that early on already. So Tony, we've gotten the plant to produce a bigger stalk, a bigger leaf, and ultimately a bigger ear size. Now, any concerns about filling that ear? And if so, how is your product going to help us make sure that we, don't just set the bigger ear, but we also fill that ear?
So I was talking about the vegetative state being improved and being able to, if the plant thinks it's happier, it will grow a larger leaf. That larger solar panel surface area of the leaf is all about filling that kernel. If you create a massive ear and a lot of kernels, but you don't have enough solar panel in there to produce the energy, you're just not going to fill it. That increased size of stalk and getting rid of that pinch point at ground level, that allows the water to move more freely throughout the plant. It's going to move that nutrition into the ear more efficiently.
So essentially what you're telling me is we're just allowing the plant to utilize photosynthesis more effectively and more of it, and we're bringing more nutrient and water up through the soil into the plant.
Yes.
Wow. Yeah, It's really fascinating. We pulled some ears. I don't know if I think I have a picture coming up here, and sometimes we see it in lengths and sometimes we see it in rows. Which one do you typically see the advantage coming in? I'm sure probably hybrids play a lot of role in that too, Tony. But do you typically see a more rounds or more length on the ears that you've been treating with ion?
Actually, we've seen pretty even in both. It is genetically tied, so it is based on the genetics. Um, that the extra rows are very beneficial. By making the plant feel it's happier earlier on and not getting that pH stress during the day, we allow the plant to throw a bigger ear. The rows are kind of key to that. But the length, we've seen length in most of them, and we've seen better fill to the tip. By being able to support the pollen growth early on when it's starting to get hot. With the heat waves we've seen recently, it's actually changing the ability to create the pollen. So they're throwing tubes, but there's no genetic material in them. So we've seen biologicals help the ability to put genetic material into that pollen more efficiently.
Interesting. Well, speaking of heat and drought, um, you know, that's been probably the biggest visual that we've seen the last 2 years in some of our plots, particularly I think of Lake Preston, South Dakota, the southeast corner of Minnesota there, where it's been bone dry with really high heat the last 2 years. But we have seen some dramatic differences between treated and untreated. And again, I'll have a picture here in a couple slides, but how is your product helping assist these plants get through those hot, dry spells and allowing us to still maximize our yield?
So by improving the stalk diameter, we're getting better water movement, but it's all about maintaining that pH in the leaves. If you're able to maintain the pH in the leaf, the plant is less likely to go into photorespiration and eat the sugars it did create. By maintaining that pH, we're allowing it to create more sugars, and it's effective up to about 95 degrees is what we found in our chamber across most genetics. So it will allow that stoma to stay open. It's not stressed as quickly. By being able to maintain that pH, it's been key to keeping it from eating its own sugars. If you can prevent the plant from eating the sugars it did create, it's putting those into the kernels. It's pennies going into your kernel every day that you're losing, and those add up pretty fast. So anytime that stoma is closed and it's stressed and pineappled, it's eating the sugar that should have gone into your corn kernel. We help prevent that by maintaining the pH.
So it's just basically going into a lockdown mode and it's cannibalizing itself.
Yes.
And I talked about this last slide, the residue management. I think we got to do more work on this to really find the value, because I don't know if anybody really knows how to measure residue management value just yet. But walk us through, what we could expect to see with ion effects helping us digest down that leftover residue.
So this one's actually, everyone can thank a farmer up by Redfield, South Dakota for this one. He has corn on corn for better than 30 years. He cuts it near the house for silage. He wanted me to put something in there that would help break down the cellulose inside of the plant. So I took some of the biologics we were playing with for an inoculant for silage pits, added it into the system, and it only works when the plant has been cut and is dying. So it will not impact the plant until it is actually starting to senesce. So when you cut the plant, it actually breaks down the carbohydrates in the stalk. So once the kernel's formed, it shuts off the ability for the rest of the plant to put more sugars in. Well, the plant is still alive and it's creating more sugars. I take those sugars and put them down into the roots by converting them into organic acids and alcohols. When that happens, it starts to break down the stalks and the residue down to just the cellulose, just the lignin. So the cellulose is actually converted into organic acids and sugars. Alcohols drop down into the soils. And when you do that, you're able to just pull apart in as little as 60 days from his silage stalks. He's able to pull them apart like a paintbrush, just with your hands. So this was one gentleman's request not to have tire poppers in his field that we were successful with.
So essentially, he's just you guys are just carving out the middle. You're just eating down the meat inside of it and allowing the outer layers to be kind of peeled back like an onion at the end of the season, and very shatterable and easy to digest down for the bacteria the next spring.
Yep. So they shatter when you drive across them. They're easier to plant into. And this was just kind of an offhand request that he made. It's like, I have bacteria that could do that, but they will only live exposed to air when you cut the plant. So it had to work, but it had to only work after it was cut and dead.
And essentially, and at some point they will go dormant or die off due to temperature, I'm assuming, Tony, correct?
Oh yeah, these guys freeze easy. And
Yeah.
then they're kind of wimps. So the wild bacteria, think of it putting a small young sheep out into the African savanna, it's going to get eaten. My bugs are wimps. They were kind of bred that way. I didn't want them to last over winter in case you change crop for next year. In rotation, I didn't want them overwintering. So I bred them to be kind of wimps, and the wild stuff out there loves to eat them.
So Tony, so you maybe added 20 or 30 or 40 microbes into the Ion Effects consortium. Some people like that word, some people don't. Why add more? What do you think? Why add more? And maybe what do you see those more microbes adding? On the label, you only have a couple of microbes, and we know that that's what you're allowed and is able to do in terms of inert versus actives and these kinds of things. But I think that it's important to think about those activities. Could you elaborate on that just a little bit?
So we found a lot of genetic variability within corn. Let's just pick on corn here. There's a lot of genetic variability. And when people pick a genetic, they're looking for a plant physical response. They don't pay a lot of attention to the biochemical in the plant impact. As long as it does this one thing, wider leaf, thicker leaf, better rib in the leaf, that's what they're looking for. And by changing the biochemistry to get that, we've found that if we have a zinc solubilizer, I don't need just one, I need about 6, because each one reacts a little differently in those genetics. And I can't go genetic by genetic. I can't go seed company by seed company developing a product. I've got to make it work for all corn. And that's why we've had to start adding more bacteria. It's just to kind of make sure that it functions in every one of the genetics we've found.
But yeah, so basically, Tony, you're saying rather than us trying to pick and choose which hybrids it's going to work on, you're trying to make your product a little bit more universal across all hybrids.
Yes.
Yeah, I think that makes a lot of sense. Especially as we're getting, wider and wider geographical area where this product's being used. So I do want to just go through the acre matchmaker real quick here, and then I'm going to just pause for a minute and let anybody else in the group ask a question. But this is kind of what we've put together on where we think it has worked the best for us. If you get it on a seed coat, it seems to have a really high likelihood of success. Strong fertility program, we've talked about that. Just getting a bigger plant with a bigger ear. We need the fertility to make sure we take advantage of that. Late foliar application is not our friend. We do not want to go out at a fungicide pass and apply this and expect to see all those early advantages that we saw, if you get it down V3 or prior. Heat and drought stressed areas has been very successful with us in terms of just, balancing and handling a lot of that stress that Tony kind of went through and how we can do that. And then the minimum till or no till ground on just helping break down that residue and recycle those nutrients back. So those are the areas. I mean, this isn't everywhere that we'd put it, but these are the areas that we think, have the strongest likelihood of success. Tony, is there any other area that you think we need to add to this list on just where it have a strong likelihood of success?
We've seen a lot of interesting results in really sandy, dry soils, loose dry soils. So by putting a bigger root mass in there, year after year, we are finding that it's increasing the organic matter and improving the water holding capacity of those dry, loose soils.
Yeah, I would counter you on that a little bit, Tony, and say that some of our biggest successes have been on the clay loams where root development is a struggle, especially early on in the season, and that's been some of our biggest wins as well. So I think you make a great point there on some of those looser, lower CEC soils, but the clay loams and the hard packs for us, where natural root development struggles, I think is an area that we've seen a lot of really nice advantages as well.
So there's a lot of same year and subsequent year impacts. So the year after you use it on corn, we've seen impacts. You can actually see our test strips the following year because of the organics left. That's kind of where I was going with it.
Oh, perfect.
So it's
Yeah, that makes sense.
year to year benefit.
Right. Yeah, if you're looking at it from a program standpoint, yep. I'm going to pause here for a minute before we move on to the trial data, because I'm going to fly through the data real quick, because we can always follow up with an email on that. But any questions for Tony on ion effects from the group?
Looks like you do have a hand raised.
Go ahead.
Hey, Tony. Minders from Southern Indiana. Kind of in relation to the soil type question, we're told by some microbiologists that we see the biggest benefit with biologics in clay environments. And I guess the question would be related to maybe weather patterns. Are there anything? Okay, so we've got 40 to 50 inches of rainfall a year. Are we going to see bigger benefits in heavy rainfall environments or drier arid?
Good question. So, Tony, the question was, are we going to see more advantage on a heavy rainfall year, or are we going to see more advantage on a drier kind of drought type of year with this product?
So the ion effects likes extremes. So it shows best in an extreme. So excessive water or excessive dry in a 40-inch clay soil, it's going to support that growth. It's gonna push more yield than biomass. In a good Illinois soil with adequate rain, you're going to see more of a yield push than a biomass push because the plant is already happy. That increase in root mass is going to get you a faster tap, better laterals out. So you're going to get less wind damage because you're going to have better structural support in the soil. In good years like that, where you have adequate moisture, good soils, good fertility, that maximizing yield is what this is going to be about, not necessarily the extra biomass and the visual response you'll get on an extreme year. Everybody wants to be able to drive by at 60 mile an hour and see that it made an impact in the field. You see that most in an extreme year, but in a normal year with good soils, it's going to push the yield rather than the biomass.
Yeah, I think that's been pretty true with our experience too, Tony. Shandy, are there any other questions on the board?
Hey, Josh, I've got a question for you and Tony on working with a fair amount of dairies over here in the west and the north. What have you seen, Tony, on just the tonnage factor from how INX FX can help digestibility, those types of things that would make for a good conversation with a very important sector out here in the dairy.
Yeah, so you're kind of leading us right into where we want to go there. Trial data wise, this is last year's data. We did 22 different data points across our LEAP plots, we call them, but there's 4 different states involved in that and 7 different hybrids. And we averaged a little over 2.4 ton to be exact. I think maybe it was a little bit inflated. We're probably closer on average over the multi-year on silage to about 1.5 ton, somewhere in that ballpark. But tonnage wise, I can talk to that it has been a beast for all the reasons that Tony's talked about on just creating a bigger plant and bigger ear. Tony, I don't know if you want to elaborate just a little bit. We have a couple minutes here before we get to Soy FX, and then we got to start wrapping up Ion. But on the base Ion FX product, not talking any increase in bacteria, but the base product, is there benefit to quality, digestibility, any of those types of things?
So on the base Ion FX, it's going to give you the increase in tonnage, and it's going to give you an increase in starch content because of the increase in overall yield. So it's going to give you a little bit better energy in your silage. It's going to give you a yield and tonnage increase. We've separated out regular Ion FX from a silage product that we'll be promoting here shortly. But the regular Ion FX will give you an increase in tonnage. We've seen an increase in milk per acre because you're getting extra starch and you're getting that breakdown that is in part of the residue management. You're getting that in your silage pile as well. So you're getting a little bit better breakdown. It works great in combination with a pit inoculant to give you a 1-2 punch on breakdown. It's that increase in starch that you're going to see from that boost in alcohol and organic acid content in the plant itself, and that extra starch.
Yeah, I think we've seen the same thing, right? So it's more silage coming off the field, obviously turns into more milk per acre just by volume there as well. So it is a big market for us. As Tony kind of alluded to, we're in the final stages of having a silage specific version of the product that will touch on some other quality things that we'll talk a little bit more about later this winter. But for now, the base product, if you're just trying to chase tonnage and a little bit of benefit on the quality and the starch, it certainly holds its weight in that arena. And as we get to our data set, this is from this past season 2022. We had 7 locations with it. We averaged just over 8.7 bushel per acre with the product this year. A lot of these locations had heat and drought stress, so obviously that influenced it quite a bit. But it was just rock solid for us this past season across our plots as a seed coat. So, Shani, one last call for questions before we kind of move on to the Soy FX. I know we're getting short on time.
Yeah, looks like I'm having. Do you see it, Michael? Somebody asked something. I thought it was you. Okay. Thoughts on applying the FX products 2x0 dribbled on the surface behind the planter closing wheels instead of in furrow?
Tony, I'll let you take that one.
All right. So my biologics, when I said they were wimps, they are, and they require sucrose to be able to live. So you will not get transfer from plant to plant, even if there's water between them. They are sugar dependent. They cannot live very long without a food source. So putting it on the surface of the ground behind without putting it either in contact with the seed or the plant, they're not going to live long enough to inoculate the plant. You won't get enough of the biologics in there to make a huge difference.
That's a great question, though.
I think that's it for now.
Perfect. And here's an example, just real quick. I promised you guys a picture. This is Lake Preston, control on the left, and the next row over was the seed treated right. You can see the difference in just how hard the control was pineappling or curling compared to the treated there on the right on a hot day in South Dakota there. And then this is down in southern Wisconsin in the Platteville area on 106-day corn. Again, just talking about that brace root development right out of the gate, the thicker, bigger stalks, bigger ear set. And I believe this one, if I'm not mistaken, ended up being close to a 15 bushel difference down there in Platteville on this hybrid. So definitely, is a beast on both silage and on yield on grain corn. So Tony, I'm going to switch gears a little bit just because we're getting short on time, but we're going to move into Soy FX. And another one that's a seed coat for us. You can use it as an early foliar or in furrow if you have the capability with beans. And again, 100-day seed viability with it is what we warranty on it. But we're going to kind of fast forward ahead here and just go to kind of an illustration that I put together that I think talks about the product in the best way. And it's this one here, but I'm going to kind of guide you through it, Tony, because I know you haven't seen this slide, but walk us through right out of the gate with this product. What we see is this additional branching that occurs on the lower axillary buds, and we kind of more or less manipulate the plant into believing those additional branches are their own plant, if I'm not mistaken. Is that correct?
Correct. So by changing the biochemistry inside the plant, that's why you don't want things to overwinter. You don't want something from a soybean to survive into corn the next year. When you're doing a successful rotation, you want the biologics that I put into these jugs to die over winter. This one causes additional branching inside of the soybean plant, but it considers the first 4 nodes on the soybean plant, you can clone an entire plant from those. So you can cut them off and regrow them and grow an entire new plant in a science lab. I trick those first 4 nodes into thinking they've been cut off and they're their own plant. So we brought in some of the biologics that give off cloning hormone, and it tricks the plant into thinking that each one of those clonable nodes is its own plant. So we can increase the pod count significantly. This is where fertility really matters. You want to make sure that you get enough fertility in there to support filling those pods. If I give you an extra 10 to 50 pods per plant, based on genetics, please feed those plants. We're going to give you extra pods per node. We're going to give you more nodes, and that branching is going to give you some spectacular results.
Yeah, so we've seen that a lot. I mean, we pulled 290 plants, I think it was last year, the year before, and I have the numbers up here. It was 23% more nodulation, but 28% more pods per plant is what we saw on those 290 plants. Now, to Tony's point, not all those pods, result in yield, right? There's a couple things that have to come into play. One is the fertility that Tony certainly talked about there, but 2 is moisture during pod fill. So we can be dry. We learned that this year. I was really nervous about soy effects this year in our plots because we were dry across most of our plot area. However, we did catch some decent rains end of July, early August during pod fill, and we had some really nice results with soy effects this year. So we can be dry early as long as we catch, in our opinion, as long as we catch moisture during pod fill. And Tony, would you kind of agree with that analysis?
Absolutely.
So again, those additional branches, they're going to throw some additional flowers and ultimately more additional pods that come off of that. Tony, where are we going to see these pods? Are they going to be, the top half of the plant, the bottom half of the plant? What should we be on the lookout for?
You're going to see a lot of mid to lower pods. So a good leggy bean. When you pick your varieties and you're going to use soy effects, good leggy beans are important. Making sure you get that head down as low as you can. So pod fill is from the bottom up. We have some fields where we went in behind the combine, pulled 4 to 5 pods per plant off, and then tested test weights on them. We had some 80-pound test weights on the beans that were left behind. I had a farmer early on in our trials, before we explained to him, you've got to go slow and lower that head. We had 15 bushel left behind, and those beans were 80-pound test weights. It was a learning experience to partner a genetic with harvest practices, but if you partner those 2 up, you can get some amazing results.
So,
and this one of the important things about this product, because it's going to set more branching and pods, you can take these thinner line beans and turn them into more bushy beans. And then also with a lot of the checks that we did this summer, and we got pictures sent to us by different people, instead of pod counts being 40 or 50 or 55, we were seeing pod counts 60, 80, 90. So a 20 up to 80% increase in pod counts. And what we like about that is that we're giving the opportunity, if you're going to foliar feed, if you can do the right activities later in the season, to hold those pods and fill them out, we set the potential for greater yield. Now you have to have all the things come together, your management practices plus rains. And I know a lot of us didn't get a shall we say, those hurricane rains late in the season, like we were accustomed to. So maybe we had some fields where some yield had gotten topped off. But this product just does things that we've not seen done before by any type of biological, that I've seen.
Yeah, I think you're there, Mike. I always tell our guys that this is probably our most unique product that we have in the catalog today. I've been in biologicals 13 years and I've never seen a biological do what this one does to a plant and change in terms of just changing the physical structure of it as much as it does. And Shandy, I apologize, I cut you off there.
No, you're fine. I didn't know. We have a couple questions. I didn't know if you guys want to go through
Yes,
those
let's do the
now.
questions. Yep.
Okay, first question is, what is the best way to feed soybeans for best yield when using Soy FX, base soil nutrition, or foliar feeding?
Tony, what's your thoughts? I have my opinion, but I want to hear yours first.
So the fastest plateau we've seen is a micronutrition. Lack of the micros later in the season because they've been compartmentalized in the roots. A good mid to late season foliar on micronutrition. I really like to see that one, especially a molybdenum. Something in there to help the movement of carbohydrate and protein inside the plant. Micronutrition late as a foliar. Mid-season foliar application. I've got one of my trials this year did an 18 on nitrogen as a foliar, and he had some absolutely spectacular results. I want to play with that a little more this year, though.
Yeah, my opinion was identical to yours, Tony. That our best results from talking to growers have been when they've gone out and done a mid to late season shot. A lot of our growers will throw some form of a potassium in with their fungicide and do a shot of potassium to really help get it across the finish line there. So we're seeing a lot of the same stuff that you just mentioned. So, Shandy, another question?
Yeah,
go ahead, Mike.
I was going to say, based upon what Tony's saying also, this is where we would like a nitrogen fixer to stack with this product, give us even a greater potential. As we go later in the season, we have the nodules lose some of their effectiveness, right, on those soybean roots. So a nitrogen fixer in there is going to keep pumping that nitrogen to the plant to give us better fill. And then Trenton's question kind of goes along those same lines. What's the good, better, best suggestions on soybeans mid and late season on the foliar feeding? And question along that line was, is foliar feeding happening with the farmers that you see that are getting these 80+ bushel per acre yields?
I think in most cases they probably are doing some form of foliar feeding. I know we do have some that will combine this with another biological, whether it be something like a terracin that helps get the micronutrients out of the soils more efficiently into the plant, or like an Invita type product that helps feed it nitrogen throughout the course of the season. So this product plays really well with our group on bundling with something else to kind of help that fertility, and the ROI is still significant enough that it pays off at the end of the season. Tony, I don't know if you have any thoughts there too.
I'm a firm believer in stacking additional biologicals, anything that's going to fix more N, especially later in the season. There's some of the nitrogen fixers out there or early season. If you get something like an Invita on there where it's doing a late season nitrogen, that is incredibly beneficial. I haven't seen it in the stuff I've been working on that it reduces the N from nodulation by stacking a biological that works in the leaves in the upper portion. We've seen a lot more yield coming out of it just because you can't get nitrogen into the field any other way late in the season.
I think, we talked about this with our group at the end of last week, not to get off in the weeds here, but one of the things that really impacts nodulation on soybeans is planting into dry conditions. It just really hurts the rhizobia bacteria on creating those nodules. And so I think, to Tony's point there, something that can help you get late season nitrogen particularly with his product, is going to be a big benefit. But I think just in general, it's going to be a big benefit next year as well. So just keep that in mind as we're starting to look at what the planting conditions are going to look like in the spring.
There's looks like Chad has his hand raised. Do you want to ask your question?
Yeah, thanks again. I've got some comments that we get from folks when we recommend foliar feeding is that their beans are going to stay greener longer into the season, which we could look at as a benefit in a lot of people's eyes. But what kind of suggestions would you have for these folks that make these comments or are nervous about trying to push soybean yields? Some of these folks are planting beans before their corn, so they're trying to go get them in wet or a little premature sometimes. But any other ideas?
So if I heard the question correctly, it was trying to prevent the stay green for too long on soybeans. Is that what you're getting at there?
Correct.
Yeah. So I'll talk from our experience and let Tony give his input, but we have not seen any additional stay green that's impacted our harvest timeline. I think we've seen healthier beans throughout the course of the year, certainly in the stressful conditions when you maybe get some leaf abscess or some, buildup of ethylene that's causing some yellowing on the leaves. But in terms of manipulating the actual timeline of harvest, I don't think we've seen any of that in our plots. But Tony, what's your opinion on that?
And anything where we've had the additional pod development early on, we haven't seen any delay in harvest. The pods are so thirsty for nutrition that it takes it all up really, really quickly. The plant really wants it, and if it gets access to it, it's going to put it right into those pods and right into your yield.
Okay, Tony, comment on your speeding up maturity here. I think that's relevant to the discussion, especially with the activity on this product. We've got a large acreage customer over in Northeast Indiana up at Fort Wayne, and he was so excited this year because his SoyFX, he was taking 3.8 soybeans out 1 to 2 weeks ahead of the other people with 3 to 3.1 or 2.3 type of maturity soybeans. So I know you've got some ideas on that.
So one of the things we found is we are getting you to R1 quicker. By getting you to R1 quicker, it's allowing you to get to maturity and R6 where you're not going to put any more pods on. We're getting that up to 2 weeks earlier. So if you've got the correct genetic in there, we can have a huge impact on your maturity at the end by getting you to reproductive stage quicker.
Yes. Okay.
Sorry. Looks like the last question is, are there gram-negative bacteria in ion and SoyFX for the N-fixer pieces?
I can't answer that one, Tony. That would have to be up to you. Yep. I'm not sure. I don't know the answer. No, let's put it that way.
So we've got gram positive and gram negatives in there. That way we try to do some broad spectrum. So if you've got so there are beneficials in the soil that will kill a lot of the gram negatives, but in soils that don't have those beneficials, you need a few in there to be able to overcome the bad actors. So you want them to be out competing in your plant something that is going to be detrimental to the plant. So we do have some of both in there just to help the overall plant health. You want beneficials to outcompete anything that is parasitic or detrimental to the plant.
Thank you for answering that, Tony. So as we look at our recommendations with it, again, seed coat has been our best response with it. We see a nicer response the later the variety. So group 2s and above, we seem to see a bigger response with that. Although this year we did have some really nice responses on a group 1. So it's not necessarily a hard set rule, but just expectations wise, the later the variety, the better the odds are in our experience. Late foliar application, again, for us, we just think getting it on early with what it does, that lower branching is beneficial. I don't think I would wait, till fungicide timing to put this down. We just haven't seen it, with one caveat, which I'll get to here in a minute. And then heat and drought stress areas, again, we can be dry early, but dry during pod fill is pretty negative for us. On that high management fertility, we got to have fertility to fill these pods. Those are your two biggest things: moisture and fertility. And the reason I said one caveat to the late foliar application is the efficacy after a hail event. So we've seen some really nice regrowth after a hail event, and this year we saw it as a foliar. Actually, we went in within 7 days of that field getting hit with hail and sprayed it on foliar, and we saw some really nice rebranching. Tony, spend just a minute on what we're doing on hail damaged beans.
Okay, so on hail damaged beans, when I mentioned the fact that our bacteria trick the clonal nodes into reforming as a whole new plant, when you get a hail event and it cuts the plant off, you are going to regrow from the cut, not from the node below like you normally do breaking apical dominance. You're going to regrow from the cut. So an ROS stress response from hail or from an animal eating the top of that plant off, you're going to get that response in the biologics. It wants to rebuild the host. It wants its children to have a good place to live is the best way to think of it. It is going to do its best to rebuild that host, and your plant and yield is that host. So we had a product called Replant, R3 Plant, that we found we couldn't warehouse enough to get on after application. So we just folded that product into SoyFX, and that's what causes that hail recovery response. And it doesn't have to be a total loss. It can be a partial loss at 25 to 28%. You can end up with a higher yield at the end of the year from a 25 to 28% loss the last week in July than you would have if you hadn't gotten hail.
Hey, Tony, could you speak to just a little bit? You and I had a conversation about a grower that had a hail event, but those beans have to be a certain stage before we can make that work. Can you speak to that a little bit?
Okay, so what the plant will do is it will regrow using energy stored in the root. So it's a really rapid regrowth. You have to have enough root mass on there to be able to support that regrowth. If you don't have enough energy storage and enough root mass to be able to feed it quickly, it doesn't have the same impact. You've got to have a good root mass. So you need to probably be right about R1 with enough root down below the ground to support rapid regrowth, because it will try to regrow that plant very rapidly. But to Josh's point, you can spray on SoyFX foliar 7 to 14 days after the hail event and bring that same response back. It's just hard to get the product that late in the year.
Yeah, I mean, our advice is get it on the seed and don't worry about it, and that way it's built in as kind of one of those little freebies again. And then just summarizing the data here, we did have one plot come in just last week that I don't have in here. So we're right at about 3 bushel per acre average across our geographical area last year, which, for the price of the product is right around a 3-to-1 ROI. So it's been a really solid product this past year. I think almost all of our plots this past year were positive. I think we did win. I think every plot we won technically this past year. So it was a great product for us in 2022. Questions, Shandy, any more questions on soy before we jump to the last one here, which is crown? And we're not going to spend nearly as much time on crown effects, just a few minutes to kind of give a quick summary of it.
Nope, looks like we're caught up.
Awesome. I know we're running along here, guys. I apologize. We'll wrap up crown effects fairly quickly. Let me switch over to a slide deck here real quick. So Tony, give the background on crown effects and why it's a different product than the soy and ion.
So Crown FX is designed to give you really rapid root growth, but I want head-bearing tillers to form. So I put a group of biologics in there that support the plant throwing a lot of early tillers with really wide roots. So this works really well in a small grain, but you don't want to use this in a soybean or a corn. You don't want a corn to have 6 tillers coming off of it within the first 2 weeks. But when you've got a small grain, closing that canopy faster, getting more head-bearing tillers is really important to uniformity in the field and that rapid canopy closing to reduce both fungal issues and moisture loss.
Yeah, and we don't have as much experience with Crown as we do with soy and Ion, but the experience we do have with it has been very nice. So again, Tony talked about that early season emergence, getting those tillers going to close it quicker, faster canopy. We've certainly seen that. Larger flag leaves, Tony, why is that important?
So a larger flag leaf is going to give you more surface area to be able to create carbohydrate and fill the kernels. So by creating, we're giving you extra inches of head per acre and extra numbers of kernels per inch. So benefit to the larger flag leaf, that's going to give you better fill and uniformity at the end of the year. We've also seen a protein increase across the board on these. You can even get that protein boost by using this as a foliar just before flag set. So just before that flag leaf comes out, that flag leaf is improving your quality of grain, your test weights, and your total yield.
So it's just a bigger solar panel again, to use that analogy
Yes.
from earlier.
Yep.
Straw residue post harvest. I know with where forages are at, that's becoming a bigger and bigger deal. What should we expect there?
25 to 41% is what we've been seeing consistently. It's all about the variety. You've got some that they're trying to keep stunted, so they're putting on PGRs to keep them from getting taller. If you're looking for good residue and good straw, the uniformity on this is making it easier to harvest. You're able to raise that head farther off the ground and leave more for straw. This one for residue management and for trying to get more organics into the soil, this product has been outstanding for that. That extra root mass that you're seeing in the picture, that extra leaf mass. One thing you'll want to do is you're going to end up raising the head, whether you like it or not, because you'll start to overwhelm the rethresh in the back. So that extra residue is holding moisture, it's holding snow, it's giving you better organics in the spring for the next crop. This is the one we're seeing the most carryover for benefit into the following year. If you do this on a small grain, you will see your test trial plots the following year in the next crop that's planted in it.
And I don't have an acre matchmaker made for this one, Tony, but give us some examples of where this product works really well, and maybe an example of where you probably would stay away from if there is one.
So we found you want to stay as moist as you can. You don't want to put this into dry desert areas. It's going to grow a bigger, faster plant. You need to have enough moisture to fill those kernels. If you're not going to get a drop of moisture after July 15th, don't use it. But if you've got good moisture in your fields, this is an absolutely outstanding product across almost every small grain we've tried.
What about winter versus spring wheat?
Winter over spring wheat. So this has been an interesting one. We had a gentleman that called up. He did a trial in Kansas for us. He is actually grazing the treated side, but not the untreated. So by doing winter wheat in Kansas, he actually got some grazing capacity out of it because it grew tall enough to have plant material there to harvest. So it's going to give you better tillering, better root development in the fall that will give you better overwinter capacity. He's going to hit it. He did it as a seed coat. He's actually going to replant it or reapply as a foliar in the spring just before flag leaf to try and give himself the benefit of the protein as well.
Mike, Brian, Shandy, any questions?
Yeah, so Tony, on the concept of if you have planted in the fall and you put it on as potentially as a seed coat in the fall to get all those benefits at that time, is the suggestion or need that because you have a lot of die-off of microbes over the winter, that you would want a reapplication, a foliar application in the spring also?
If you're looking for the protein boost, it's going to give you the extra tillers. It's going to have given you the extra root mass. So you will get a benefit the following year.
All right.
But if you're looking for that protein boost, you're going to want to reapply as a foliar in the spring.
And that's at what time?
Anytime prior to flag. You've got a
Okay,
big
and-
window there to apply it.
Right. And so, but we would not indicate, we want to be very clear on a rec. You would not put this material in with your 28% or your reapplication early. I'm sorry, your liquid 28 or 32 application early.
Nope. You would want to put this on. So if you're looking just to improve your protein, and a lot of people aren't paying for that extra protein, it's just to help reduce the risk of that dockage. Everybody I talk to right now is saying, well, they don't pay for protein, but they do hit you for dockage if you're low.
Right.
So
And quality, if this helps get you quality versus less toxins and so on, then you're going for the ability to be able, sometimes even just market your product, your wheat.
Yes.
If it gets bad enough, in a sense, or to get at the better quality metric and not get docked. So that's great comments for guidance on that. So if someone didn't get this on their wheat last fall as a seed coat, and we're going to go into the spring and it's going to be spring green up time, then just specifically go back one more time and say, why would they put it on in the spring? Can you still get a tillering effect or greater flag? I mean, so what's my benefit if I didn't get it on a seed coat?
You didn't get it on a seed coat, this is going to help you get better root establishment. If you had some winter kill issues from this cold, if you didn't get the snow before the cold, this is going to help fill out the tillers and get your canopy closed faster. Spring application into a winter wheat is going to be beneficial. It's going to close up that canopy faster. It's going to help with your moisture retention, and that quality piece is going to be a big deal.
Thank you. Any other comments?
There's a question. Can I run Crown FX in furrow with starter fertilizer? Will the starter fertilizer have potential to kill the microbes before they get into the plant? And then with spring foliar application, are there certain classes of adjuvants that can help with effectiveness?
All right, so from an adjuvant standpoint, we have bacteria built into the bottle that will help work as an adjuvant. We don't recommend the use of those with the product directly. You can, but it's not necessary. It's not required. Also with a starter fertilizer, for the starter fertilizers the last couple of years, it's like going down and buying from your local drug dealer. It is amazing what this stuff is cut with. It's quality of the salts that really matter in a starter fertilizer. The more salt and the more cutting agents, for lack of a better word, that's in that starter fertilizer, the more issues we've seen with die-off of biologics. If you use a dosatron system to inject and not have it in the tank with it, we've seen less of an issue. But fertilizer quality is the key to this one. That's why we don't really make a recommendation on fertilizers. The quality is so all over the board based on where you get it from and the time of year you buy it. That's why we're kind of looking at trying to do it with slightly less stressful additives. And cereal rye, it works the same as it does in wheat, to answer that question. Rye, barley, teff grass, wheat, triticale, we've seen it across the board on small grains.
Yeah, we did some trials in Colorado and had some really dramatic results out there under pivot as well. So, and to Tony's point, we're in the same camp as him. We're recommending on biologicals in furrow. I mean, if you're going to spend the money on the product, make a long-term investment and just buy a dosatron. They're not that expensive. I think you can get them between $1,200 to $1,500 depending on where you get them from. And it can save you a lot of headaches. So I would highly recommend having your growers look into those. But Shandy, any
Looks like
last
we have
questions?
one more-
Josh, go ahead.
One more question. Looks like Chad has his hand raised.
Go ahead.
Go ahead, Chad.
Yep. I was interested to know, Tony, is there any compatibility with fish hydrolysate? We find that there's a lot of micronutrients in most fish products as a micro pack, as well as your calcium. And, if you've got a sulfur-based stabilization there, is any of that a benefit or a harm to either of the three products?
Actually, we've seen some pretty cool results with fish emulsions. You want to do a jar test with anything you're going to add it to, but we've seen some pretty cool results. Those fish emulsions really do support biologic activity inside the plant. Co-mingling in the tank to apply it, there's been a few issues just because water chemistry is different, but fish emulsion within the system as an additive post or pre, we've seen some cool results on that.
Okay, how about water pH or where is there a best practices for our water conditioning?
So we actually use an API water conditioner that you buy at the pet store. It's a heavy metals and chlorine remover. A lot of water in the springtime is coming from surface water. They switch from wells to surface water in the rural water systems, and they put a lot of chlorine and fluoride in there. I do recommend, if you're going to use treated water, that you use that heavy metal and chlorine reducing pond product from API. It's cheap.
Thank you.
Looks like we have one more question. Oh, Brian, you have a
Hey,
question?
Josh, this is Brian.
Yep.
Yeah, this is Brian. Cover crop. Cover crop's been a huge issue. Carbon sequestration, everything else. What about cover crop? Any of these products?
Yeah, Tony, is there a fit for any of these three products we talked about today on cover crops? I have an additional comment I'll make when you're done with this, but.
Oh, absolutely. If you're throwing a legume in the cover crop, throw a little soy FX into the mix on the seed. If you're using a grass, throw on that Crown FX. It will give you faster, better roots, more cover, better organics. Absolutely use this with a cover crop. It is worth the investment.
Yeah, I would agree with what Tony just said there. And then I would also add on to that. I talked to a professor from University of Wisconsin Extension 2 weeks ago at a conference on cover crops. And she made the comment that your first 2 to 3 years, your yield's going to be a little bit out of whack on cover crops because you go backwards a little bit. And I asked her why, and she said, "Well, it's your nitrogen to carbon ratio gets a little crazy for a little while there." So, and she said the problem is a lot of people in Wisconsin can't go out and just throw more nitrogen down because they're under management plans. So Envita or any other really good end fixing product would also help in those situations to make sure that you're getting as much nitrogen to the plant to balance out that C to N ratio without necessarily having to put it down on your nitrogen management plan. So, as Tony talked about earlier, stacking is a great thing to do as well. And if he can get you more plant surface and we can get you a little bit more nitrogen to support it, those two things together work really well. Any other questions?
Looks like that's all for now.
Hey, thanks a lot. I just want to make sure we hit on that because I don't want people to think we had talked a lot about spring applied only. So I want everybody to understand that this is a season long application. So thanks for that.
No, that's a great question. So I know we ran long today, but I thought while we had Tony here, because he's hard to get on these calls, he's got a very busy schedule. I thought it'd be beneficial to really do more of an interview with him versus me PowerPointing you to death. So I appreciate everyone willing to stick around on it. And Michael, I'll let you kind of close us out.
Sure. We really appreciate this, especially with Tony coming on and his insights. Want to let you know that we are available to do these kinds of webinars one-on-one with customers, and we can bring Josh and Shandy and Tony in as time allows. So I'm going to turn off the recording at this time, and we appreciate your participation. You're welcome to stay on and chat here a little bit if you have any further questions.